order? No, right, okay. No, you don't have to put it back in order. That would be crazy. Okay. Okay, don't forget your seating chart. Don't forget your seating chart. They're alphabetical. Is that right?
Oh, I didn't realize.
They're alphabetical. I try. I don't always get it, but I try. Okay. I need to find where we were. Where did we end?
Yeah, I knew that was gonna happen.
They're alphabetical. We were talking about the alpha helix. Yeah, I think we were here.
I have a question. We're gonna see like who got right or wrong. Where's it at?
Last sheet. It shows you like here, it says what you did. Oh no, primary is the key. What you put, how many points you got, and was it correct?
Everyone. No, that's okay. That's okay. So y'all, if you're looking at your exam right now, the last page is where you'll see what the correct answers were for every single question, what your grade was, right? All that stuff is on the very last page. That way I can put them up here and you can find them without everybody seeing your grade, right? So I try to give you a little bit of privacy. I try. So when you're done in here, before you walk out the door, okay? Doesn't have to be back in alphabetical order, but just in here. And if you want to come see him at any time, you can come to my office and you can come see him, okay? Questions? All right. Exams are here, they're alphabetical if you want them. Okay, so the grades are posted to Moodle. So your Moodle grade should be updated with your homework. All homeworks are updated. The exam grade's updated is as updated can be as of right now. Any questions about that? Okay. Questions about the exam? Okay. Questions about the exam?
So if you have a particular question, we can see you ask.
Yeah, absolutely. Absolutely. Yes, sir. Number 26. What is 26? Okay, what kinds of amino acids are most likely to be found in proteins spanning plasma membranes? So if it spans the plasma membrane, it's embedded inside of what? Phospholipids, some kind of lipids, right? Lipids are those phospholipid tails are what? They're non-polar, so they are hydrophobic. So hydrophobic amino acids want to hang out with the hydrophobic fatty acid tails. So the majority of the protein is going to be hydrophobic, right? Maybe the tips, the ends of the protein that are exposed on either side will have some hydrophilic residues, but basically the middle is going to be hydrophobic. Okay. Y'all did really good on the problems. Y'all have to give me a little more on discussion. I'm looking for a little more. Most people did pretty good, but some people just need to give me a little bit more. Right? Yeah. What is number 12? Under which conditions would a biological reaction spontaneously proceed if delta G is greater than 0? If delta G is greater than 0, it's what? Non-spontaneous in the forward direction, right? So where's my pen? Here we go. So that's saying like A plus B goes to C, right? And it's non-spontaneous in the forward direction currently. So what can we do to make this reaction proceed spontaneously? Well, basically what we want to do is remove C, right? So if we have a lower concentration of product than reactant, what kind of a shift are we going to have? A forward shift, right? So if the reactant concentrations are greater than product concentrations, that would be a forward shift. So that's Le Chatelier, right?
But, even if it says proceed to reactant,
Under which one?
well,
Under what conditions? What do you mean proceed to reactants?
that's what the question says.
Oh, under what conditions would the reaction spontaneously proceed to, oh, I wrote that wrong. I wrote that wrong. I'll go back and give y'all credit on that one. I wrote that wrong.
So it is Le Chatelier.
So it is Le Chatelier. I wrote that wrong. Greater than the product. Yes. Yes. So I'll give you 2 points on that one. I'll just give everybody 2 points on that one. Okay. I can go in and easily do that. No big deal. That's good. Y'all always ask. I wrote that wrong. Yeah, ask. If one person has a question, other people do too. Go ahead. Brooks? Is that Brooks in the back? Okay. DNA strands containing 20 bases, 40 bases, 60 bases are denatured, and the results are on the graph. Identify the curve from the 20 base pair strand. So the smaller the piece of DNA, the easier it's going to be to denature, right? So A is the one that's denaturing the easiest at the lowest temperature. C is denaturing at the highest temperature. So the shortest strand is going to be A, the longest strand is going to be C. Does that answer? Can't see Brooks. Yes? Okay. All right. Good? Okay. All right. So take out your notes. Question? On 12, that's what I said. It's wrong. It's wrong. Okay, so if you had A plus B goes to C, right? If you want to shift the reaction in either way, whatever you want to shift it in, you remove what you're going to, right? If you want to go to reactants, then you decrease this concentration and you'll shift this way. A plus B goes to C. If you want it to go this way, you remove product. So it's all about everything's at an equilibrium. So if you change the equilibrium concentrations, then you can shift a reaction one way or the other, whatever you want it to do. But it just depends on what the delta G value is for the forward or the reverse reaction. So I wrote the question backwards, is what I'm saying. Does that make sense? Okay. Okay. It happens. It happens. The answer is it's an incorrect. There are multiple responses that are correct because it's not right. So the answer marked on there that should have been correct was B, but B would only be true if it said under what conditions could a biological reaction spontaneously proceed to products. So if you change that word to products, then B would be the correct answer. Y'all see that? Okay. Okay, so that's my fault. I'm gonna give everybody 2 points. There was already a little curve on there too. So you'll notice that what was on your paper is a little higher in Moodle. So there's a little curve. Okay. Good. All right. Okay. Always ask. Always ask. All right. So we were talking about secondary structures, right? So when we talk about proteins and protein folding, what's the organization, right? First we have primary structure. What's primary structure? The sequence of the amino acids. What's secondary structure? Beta sheets, alpha helices, beta turns, and there are also loops, but your book doesn't really talk about that, and that's okay. All right. So what we're doing is we're talking about what those secondary structures are. That's kind of where we ended off, right? In the right place? Okay, sometimes I get sidetracked. All right, so this is an example of a ribbon diagram of an alpha helix, right? And we said the ribbon represents what? That red ribbon in the picture. The backbone, what's the backbone? NCC. That's right, NCC is our backbone. And then what are those things that are sticking out? Those are R groups. Those are all R groups. Now I have a very important question to ask you. Are all interactions within a protein between R groups? No, they're not. Where else can they be between? The backbone, right? Because the backbone has what that can participate in hydrogen bonding? Tell me about this NCC. It's not just a nitrogen though, right? It's a nitrogen that has at least 2 hydrogens attached to it, right? So I'll just put 1 and just not label it, right? Because it kind of depends on if you're at the end or if you're in the middle of the molecule. But is nitrogen an electronegative element? Remember, hydrogen bonding is no fun. So it can be a hydrogen bonded to a nitrogen, a hydrogen bonded to an oxygen, a hydrogen bonded to a fluorine. So I have a nitrogen with a hydrogen. What can that be? A hydrogen bond. Donor or acceptor? If it's got the hydrogen, it is the donor. Okay, so what can be the receptor? What's on this carbon? We just say NCC, but that's not all it is, right? What's bound to this carbon? We have oxygens, electronegative oxygens, right? And we just do COO minus, right? That's our kind of shorthand for it. But this is the acceptor, right? So we can have these hydrogen bonding occurring in the backbone. So even though we're showing the side chains in this picture, that's not what holds an alpha helix together. What holds an alpha helix together is the actual residues on the backbone interacting. So let me show you sort of what that looks like. So here's the same sort of alpha helix, and all we're showing now are the atoms that are involved in the actual ribbon. Oh, yes, we need to. With all the tests, I forgot to start it. Let me do that. Oh my gosh. Yes, I'm going to tell you. Let me log in so that I don't type in my password wrong 6 times. Because when I'm trying to think and type a password, you know you get it wrong, right? Thank you for telling me that. All right, let's see. And it just takes a second to load. All right, so let's see. What was the joke? What was the joke today? Oh, why didn't the skeleton go to the prom? He didn't have the guts. Yeah, it was okay. It was okay. Charles was like, what's the prom? I was like, oh, I'm not ready for this. It's like, it's a really great party. It's okay. Right? Okay. All right, there we go. Thank you. Starting class now. Thank you. All right. And view full screen. Okay, so these residues right here, if you start here, this is the N-terminus. So all these atoms here are part of the ribbon backbone, the NCC. So when you see the little black parts, that's actually the R groups. No. Secondary structure is always going to be between the backbone. Tertiary structure is going to involve our side chains. So secondary, that's a really good designation. Secondary structure are interactions from backbone to backbone. whereas tertiary are R group to basically anything. So here's our same helix. You start at the N terminus, and do you see how it kind of wraps around? It's kind of hard to track with all those atoms, but do you see how it kind of wraps around and it goes back and forth? So, you know, if you have something that's like a shoelace, right, you can stretch it out. But if you turn that shoelace into something like a phone cord, oh, when's the last time you saw a phone cord? Been a while, right? But you know what I'm talking about, that coil. That's a helix. So if you take a phone cord, right, it's really, really short, and then you can stretch it out, right? So the residues that are on an alpha helix are really close together. This is the stretched out version of that phone cord. This is the coiled version of the phone cord. So if you look at the carbonyl, right, the oxygen double bonded to the carbon, this is the oxygen that's carrying that partial negative charge. It's going to interact with a residue that is 4 residues away in a hydrogen bond with the hydrogen bound to the nitrogen, right? So that looks like a really far distance, doesn't it? But is it in actuality? No, it's really close, because you had to go 4 residues to come back to that same spot, but just 1 coil down. And that's where you're making those hydrogen bonds. So you have hydrogen bonds throughout this entire alpha helix that's holding that structure together. So alpha helices are very, very stable structures. And proteins will spontaneously, once they're made, make alpha helices because of these hydrogen bonds that occur. It makes them very, very stable. So look at your residues. If you wanted to count your residues, the easiest thing to do is to look at the R groups, right? So this one would be any given residue. So they call it N. Then count 4 down. 1, 2, 3, 4. This is the nitrogen that starts that residue. Because remember, your NCC, right? That's 1 residue, 2, and then 3, and then 4, right? So you see how you're 4 away. The last one, right there, okay? So all of those interactions are between the backbone. Now, if you have this repetitive dipole going on, right? Because we just said that in order for hydrogen bonding to occur, what is this hydrogen carrying? A partial positive. What's the oxygen carrying? Partial negative. And the way that these line up within an alpha helice, all the dipoles line up the same direction. So now what do you end up with? An overall dipole of the entire helix. So you end up with a helix that has a partially positive charge towards the N terminus and a partially negative charge towards the C terminus. That's not really stable. So how can proteins overcome that? I don't know, it's like a negative, right? We said alpha helices a lot of times are going to be like, for example, embedded in a plasma membrane, right? It's going to have those hydrophobic residues, and it's going to basically be embedded so that it can be protected. But if this is a membrane, right, here and here, and you have all of your fatty acid chains, right, like this, what part of the protein would be sticking out? The charged residue, the charged ends. So then what happens? What can it interact with? HOH, right? Water molecules. So now you can negate some of those negative things, right? Same thing is true for soluble proteins. For soluble proteins, you can have a core of the proteins that are hydrophobic, but the ends of the helices can be exposed to the solvent.
Can you explain how it overcomes those, like the charges? They said, like,
So how to overcome a charge is to have it interact with something else that's charged, right? So one way that you could do that is the ends would be exposed to solvent, would be exposed to water, right? That would negate the positive charge on the N terminus or the negative charge on the C terminus. Either way, water can interact with it, right? So it can interact with it whether it's embedded in a membrane or whether it's soluble. It's just that the ends of the helix are exposed to water. Okay. You could interact with other molecules that are charged. Water is not the only thing that can interact with a charged portion of a partially charged portion of a protein, right? What's the charge on phosphate? Negative. So what end do you want to associate with? The N terminus or the C terminus if you're a phosphate? The N terminus, right? The opposite charge. Cations would be good to interact with what N? N or C terminus? C terminus, right? The other thing that the protein can do, and your book does talk about this, is that you can put at the ends of these helices charged residues. So what are our charged residues that we might find at the positive end? We want to find the opposite, right? So we're going to find what kind of residues here? Right here at the end, we would find negative side chains, right? Or residues, however you want to say that, right? What are our negatives? Well, you can, right now, you can have this out in front of you, whatever it is. What are your negatives? So you want aspartic acid. Oh my goodness, yes. And right now, what about here on my negative? What am I going to have for residues? I want lysine. What else? Arginine. What else? Histidine. And hist can go either way, but you could put a hist there. That makes sense? We're just trying to balance that charge out. And there are multiple ways that the cell can do it. Okay, here's our second major class of secondary structures, beta sheets. So beta sheets are, again, a very highly structured secondary component of protein structure. So if you look at a beta sheet is more like that stretched out foam cord. Those residues, do you see how farther apart those residues are in the beta sheet versus you go back to an alpha helix? Do you see how tightly packed they are? They're coiled one on top of the other. Well, here you can have a whole lot more space. And so you end up finding residues with larger side chains here in the beta sheets than you do in the alpha helices, right? Because you can fit them. It's just a space thing, right? Now, if you look at this in any given ribbon diagram, do you see how it looks like an arrow? Okay, the flat portion is the N terminus, the pointed, arrow end is the C terminus, always. And that's going to be really important because you can have some beta sheets that line up next to each other and they make these long sheets. And so there are two different ways that they can get together. They can get together parallel or they can get together anti-parallel. And they each have different characteristics. So if you look first at a parallel, right? Do you see how the arrows are pointing in the same direction? So the N termini line up and the C termini line up. If you do this backbone, NCC, NCC, the nitrogen should line up, the alpha carbon should line up, the carboxyl carbon should line up, right? Everything lines up. Do you have hydrogen bonds that end up in a perfect arrangement? No, they're kind of tilted, right? Whereas if you have something that's anti-parallel, do you see how we start with N here and we go NCC? On this one, we go NCC, NCC. So they're swapped, They're going opposite directions. Do you see how nicely these line up? So what you're looking at is a picture of the same basic width of a beta sheet. So how many hydrogen bonds are here? 3. How many hydrogen bonds are here? 4. Which one do you think is more stable? Anti-parallel is more stable. Ooh. More stable. And therefore it's more prevalent in proteins in general, right? We want more stability. And so we end up seeing proteins spontaneously fold into more anti-parallel sheets than parallel sheets, right? But when we look at the actual beta sheets, what we don't see is a flat surface. What we actually see is that the molecule kind of does a wave, right? Now, if you're into sewing, you know how to sew pleats into a skirt, a pleated skirt, that's what it looks like. That's a lot of what it looks like, but most people don't know how to sew. But that's what it was named after. But it looks like a wave, kind of looks like a wave, right? So it just gives them a little bit more free movement, free range of movement for those really big side chains. Okay, so now if you have multiple of these beta strands making up a beta sheet, how are you going to connect them? You have to have some kind of a region that will connect it, and it will have certain properties. So if we look, this is called a beta turn. So it connects one beta strand to another beta strand. And once you have all those beta strands together, we have a nice beta sheet. So the beta turns are much more unstructured. And we tend to have very small amino acids in these beta turns. So what are my small amino acids? Glycine, what else is also small? Alanine, right? Glycine is what for an R group? Just a hydrogen. Alanine is a methyl group, right? So you have to, this is a pretty gosh darn sharp turn to be found inside of a protein. So if we have a big like phenylalanine or something like that, are we going to be able to make this turn? Most likely not. So a lot of times you can look at a protein, and if you know what kind of secondary structure it has, you can predict its primary amino acid in general sequence. So there are a couple of different ways that you can have a beta turn. If you look, remember that our peptide bond is locked into place because it has resonance, right, with that carbonyl carbon. So if you look at that carbonyl oxygen, there are 2 different orientations that it can have. It can face inward, so it can face between the 2 beta strands, or it can face outward into the rest of the protein, but doesn't really make too much of a difference. Okay, so we have beta helix, we have beta strands, we have alpha helices, we have all of those secondary structures. Now, the next step is tertiary structures. So tertiary, go ahead. That's okay. This one.
For type 1, because it's a straight hydrogen bond, would that mean that type 1 is more stable than type 2?
It depends. It depends on the protein that it's inside of. There are just multiple orientations, but whichever orientation it is, it's locked. It can't swap back and forth between type 1 and type 2 because that carbonyl bond is in hybridization with the nitrogen, right? The carbon, the nitrogen bond. And so you're stuck with one or the other. It doesn't make that much of a difference. But the point is it's stuck. It can't change that. Okay. So tertiary structure. Tertiary structure looks at one polypeptide chain. Is every protein one polypeptide chain? No. Are some proteins one polypeptide chain? Absolutely. So some proteins, their structure is only defined by secondary, tertiary, primary, secondary, and tertiary, right? At tertiary, you stop. If you're one polypeptide chain, you're done. But if you're multiple subunits, we're going to have to go one more. Okay, so our tertiary structure is all of the atoms that are involved in one polypeptide chain. So it kind of looks like a mess, right? It really is really very tightly packed, but we're looking at every single atom. Now, when you look at every single atom, we took a snapshot of this protein. What's really important to remember is that proteins are always kind of moving. They have lots of flexibility in those phi and psi bonds, right? There are only certain angles that are okay in phi and psi, not all of them are. But because you have rotational movement in those phi and psi bonds on either side of the alpha carbon, remember how we said the carbonyl makes the peptide bond locked, but then on either side of the alpha carbon, you could have rotation. Remember that? So that gives the protein flexibility. Is protein flexibility important? For what? For what? Sure, binding to something, right? If it's an enzyme, it's got to conform to basically the substrate that it's binding to, and then it has to change shape when you get a product in order to kick out the product, right? There's all kinds of reasons that we need conformational changes in the protein. So when we talk about tertiary structure, it includes our secondary components, right? Our beta sheets, our alpha helices, and the position of the side chains, right? We showed that they were there in an alpha helix. We didn't really show them in a beta sheet, but they're there. And those interactions will help your tertiary structure. So do you have an ionic interaction, right? What's an example of an ionic interaction that could occur within one polypeptide chain? Remember, ionic is a positive and a negative. So give me a positive side chain. Lysine, right? Interacting with a negative side chain. Aspartic acid, right? A positive and a negative. And that helps that tertiary structure to give it your overall three-dimensional shape of that one polypeptide. Okay, so that's an example. They call that a salt bridge, right? Hydrogen bonding from one side chain to another side chain. Can we have hydrogen bonding in side chains? Absolutely, absolutely, right? So what's really cool is that you can go to what's called the protein data bank. And the protein data bank is where scientists will deposit their data that shows the atomic coordinates of all the atoms within a particular protein. So you can pick your favorite protein and you can go to the protein database and you can see its three-dimensional structure and the locations of every single atom within that molecule. Why do we care? Well, let's say that you get a mutation in a protein, right? And it leads to some genetic disorder. Well, as a scientist, if I can understand why this protein is not working, I might be able to design a treatment for it, right? So I can actually model in the protein data bank my mutation and see what happens. Does the protein not fold right? Does it disrupt an alpha helice? You know, like, did I lose a salt bridge? All those things will give me information so that, and then I can design something to treat the patient. Okay, so when we look at 3D polypeptides, there are some common classes within tertiary structures. So you could be all alpha helices, right? This is the first one. Do you see how I basically have these loops that connect alpha helices together, right? So that's one class. One class is predominantly alpha helix, right? What is B? Predominantly beta sheets, right? Y'all know what that is, B? It's a beta barrel, and what protein have we talked about is composed of a big old beta barrel? Green fluorescent protein, GFP. And that's the chromatophore right in the middle. Okay. In C, you can have a mixture where you have both alpha helices and you have beta sheets. Or you can have a protein where you could basically almost split it in half, and half of it are alpha helices and the other half are our beta sheets. Whereas this one is more of a mix. Right? So they can be equally distributed, they can be kind of all over the protein, or you can have specific domains. This domain is all alpha helices. This domain is all beta sheets. Okay. So once you get into these tertiary structures, there are some common protein folds that you're going to see. And the common protein folds will do particular functions. So you're going to, if you see this fold in a protein, you can basically predict its function, right? So if we look at cytochrome B's job is to bind to heme. So if you look, this heme group is embedded into this, basically a pocket of alpha helices. And these alpha helices surround the heme group so that they're protected, the heme group's protected from water. So if you look at the space filling, it looks like there's a lot of space here, but if you look at the space filling model, you can see that it's pretty tightly protected. So if you look at the structure, you can kind of predict the function. Another common one is the Greek key fold. So this one is beta strands that are linked together. So you can see here, you start with the N-terminus, right? And these beta sheets are all going to line up and then kind of twist in 3D. So they all follow that kind of maze structure. And then in 3 dimensions, they get kind of twisted, right? But all I'm saying is that there are multiple common tertiary structures that we find in proteins, and we can use those common structures to predict functions, right? You can say, okay, I'm studying this protein in mice, and it has this particular domain that gives it a function. Well, is there a protein like that in humans that gives it that same function? Or vice versa, you know, I want to study this protein that's mutated and hurting people. Can I find one in mice that's similar? And you can look for similarities in tertiary structure. Rossmann fold's another one. All we see here are alternating beta sheets and alpha helices, right? Alternate, alternate, alternate. So the example of this one is that these are proteins that will bind to DNA or RNA. So they'll bind to nucleotides. So you can predict which protein is going to bind to nucleotides. There's another barrel, but in this case, it's a barrel that's composed of alternating, right? These are alternating, but it's not necessarily a barrel. This Tim barrel fold is the alternating beta strands and alpha helices, but it ends up forming a barrel. And a lot of times what you do is you see these in metabolic enzymes so that your substrate comes in and we link it to the next protein and it goes through the next protein in the complex. And it's a linked metabolism, right? So it feeds from one enzyme to the next. So you see that common in lots of metabolic. Firm domain is one that's very common in cytoskeletal proteins. Cytoskeletal proteins will have these domains that are really, really common, but they only have like a 30% amino acid homology. So the sequence isn't necessarily the same, but the secondary and tertiary structures are. And so that's how we can tell that they have similar functions, even though their amino acid sequence is not necessarily exactly the same. Okay, so those are kind of like weak, all those weak interactions that hold our tertiary structures together. But we do know that we can have some covalent structures, covalent bonds, stronger things that can hold a tertiary structure together. And one of those is a disulfide bond. So your disulfide bond is going to be forming between what 2 residues? Cysteine, right? Cysteine has that reactive sulfur. So when those 2 are oxidized, then they are forming the disulfide bond or the disulfide bridge. Some places say it as a bridge, some places say it as a bond. It's a lot of times found in smaller globular things like toxins and growth factors. But there's another cool thing I'm going to talk about disulfide bonds when we talk about quaternary structure. So some proteins will stop right there at tertiary structure, right? They're complete at a tertiary structure. But a lot of them require more than one subunit to be functional. And not only do you have to have more than one subunit, but they have to be arranged in the right orientation. And so you can have, remember we talked about this before, multiple peptides of exactly the same gene. What do we call that? Multiple copies of the same polypeptide. What do we call that? Right, it comes from the same gene. So let's say you have 2 copies of the same gene. What do we call that? Same homo 2 dimer. What if it is 2 copies of a different gene or polypeptide? However you want to think about that. Right? Like the red one is one polypeptide and the blue one is a different. What do we call that? A heterodimer. So we said homodimer is the simplest quaternary structure. 2 identical came from the same gene. And then we said heterodimer is subunits from different gene products. What if you had 3 gene products? What would you call it? Of the same? A homotrimer, right? You remember all your prefixes from like naming covalent compounds, right? So what if you had 6? A homohexamer, but usually they're not, usually you don't have them all the same, but it can happen. But just all you have to do is look at those prefixes to know how many subunits. Okay, so a lot of times when you add subunits together, what you end up getting are functionality increases, more things that the protein can do. So a lot of times those portions of the peptides can be phosphorylated. They can be modified. And what does phosphorylation do for us? That switch, that on-off switch, right? So if we add subunits that can be phosphorylated, that can be dephosphorylated, now not only do we have a functional protein, but we have a functional protein that can then be regulated. So it gives us a lot more ability to control our proteins inside of our cells. A lot of times too, you can have a multi-subunit complex, right? This a lot of times happens in metabolism, where they could operate individually, but these peptides are linked together because you want A to go to B, B to go to C, C to go to D. And so we're going to link all of these metabolic enzymes together, and we're going to see this later in the semester. And then your product is going to flow very nicely from reactant to product, right? So you have that nice sequence of events. So quaternary structure helps us out a lot in that. So you're going to see some nice examples of proteins that have multiple subunits, and we'll talk about a few of them here. Keratin is an example. So keratin, where's keratin found? Hair, nails, horns, right? All that kind of stuff. So you have 2. So what do we call this? If we have 2 and they're of the same thing, a homodimer, and they are right-handed helices. They're not left-handed. These are right-handed. And so if you look at these, I'm going to tell you a kind of a funny example. You know, they're really, really strong, right? Things with keratin in them are really strong. So why are they really strong? And one of the reasons is that these coils are actually linked together with disulfide bonds. So that's a covalent linkage there that gives it that strength and stability. So why is it, if they're so strong, take hair, for example, why is it that we can go to the hairdresser? I don't like my straight hair. I want curly hair. What do they give you? A perm, What's a perm? This is your hair. When they give you a perm, they're going to break those disulfide bonds. So what kind of an environment are they going to, what kind of chemicals are they going to put in your hair? Reducing. Yes, y'all with me today? So what happens then? Well, you don't have to get a lot of perms, but if you're going to break a disulfide bond, what do you need? A reducing environment, right? Okay. So we have our two strands, right? And they're both like this. So now instead of being a disulfide bond, this little ball is an S, right? It's two S's. So instead of having that, now we have SH, right? Ooh. On each of them. They're no longer linked together. Now what do they do to your hair? They put, well, if you're trying to create a curl, they put it in curlers, right? They curl it around something. So now the strands end up looking more like this, right? You have those two interlocked. So now instead of binding to the other strand that's next to it, what actually ends up happening is you have these sulfide bonds that form within one keratin complex. So now when you wash out that chemical, you'll keep your curly hair for a while. So it's all about making and breaking disulfide bonds. I thought that was kind of cool. Okay. Silk is another one of those super strong polymers, right? Y'all have heard that? A spider's web, the silk in a spider's web is stronger than steel. Heard about that, right? It's not as flexible, but it's stronger. So it's actually lots and lots of those beta sheets laid one on top of the other and packed really, really, really tightly. And so that tight packaging leads to lots and lots of interactions. And so because of all those interactions, then you end up with a molecule that's very, very strong. So that's why silk is really cool. Collagen, right? Collagen found in your connective tissue, found in your cornea of your eye, all those kinds of places, is another fibrous protein where you have multiple strands that are linked together that give that strength, right? All these molecules that we're talking about have multiple subunits, and those multiple subunits are linked together, and that linkage gives them strength, right? So a lot of times we gain a functionality by having additional subunits inside of our proteins. Another way that you can have multiple subunits is if you have globular proteins, right? Everything we talked about before was kind of stringy looking, right? Now we're going to talk about globular proteins. Globular proteins, if you take individual monomer subunits and you put them together, you're going to introduce a new shape. So in this case, right, this is a homododecamer. So dodecamer means we have 12. We have 12 of these monomer subunits, and these 12 monomer subunits hooked together in a ring. This is a protein from a bacteriophage. And so what actually ends up happening is the virus is making its DNA, and it's made a capsid, and it needs to basically inject the DNA into the new viral capsid. And so this column, this globular or circular thing, threads the DNA into the capsid of the virus. It's really kind of cool. Everything is cool. What am I talking about? One that's really important in the news recently are immunoglobulins, right? Have you heard about this? If you get coronavirus, what is one of the treatments that they are giving you? Antibody treatments, right? So what are antibodies? Antibodies are multiple subunit globular proteins. So when we say multiple subunits, these are not two of the same. So do you see the blue? The blue is called the heavy chain. So it's very long, right? It goes all the way from one end to the other. The red, can you guess? The light chain. So the light chain stops about halfway through. So what's really interesting about antibodies is antibodies have two basic domains within them. So do you see this portion is very straight and this portion looks very wiggly, right? Okay, so the bottom portion does not vary. It's the same all the time. But this top portion is the variable region. And that really makes sense because you need to keep the same structure, you need to keep the same shape so that your antibodies all work. But do we want all antibodies to recognize the same antigen? No, otherwise your immune system would not work, right? We need to make antibodies that are unique to certain antigens. And that is the variable region. The variable region is where the antibody binds the specific antigen. So if you're looking at this picture, what connects, what holds our, let's say, heavy chain and light chain together? What do you see in that structure? I see S and S with a link together. So what is that? That's a disulfide bond. So disulfide bonds are holding together, right here, our heavy chain and light chain. It holds together our two copies of our heavy chain and the other heavy chain and light chain. And a lot of the discoveries about protein structure can also be done by using proteases. So if you give just a little bit of a protease to a protein and then look at the size fragments you get on a gel, you can start to learn things. So in this case, what they did was they took a protease that cleaves the protein right there. And so you end up with two of these part variable, part invariable regions, and then you get the bottom, which the top they call them the Fab fragments, and then they call the bottom part the FC fragments. But you don't have to know the names of those, but you do have to know heavy chain, light chain, and where the variable region is and what the variable region can do, right? And if you didn't have multiple subunits, you wouldn't, this wouldn't work, right? So anybody know how they create the antibodies for coronavirus treatment? They take B cells from people who have been infected and they isolate them in the lab so that you basically have a single well with a single B cell in it, and the B cells make antibodies, right? And so then they test the antibodies that are made by each of the B cells and finds which one is most efficient at binding to the coat protein on the outside of the coronavirus. And then they say, oh, this one had the best binding. So I'm going to amplify this one and make it produce lots of antibodies. And that's how they make the antibody treatments. It's really cool. Really cool. All right. Finally, we get to be getting to the end. All right. So you have to think about, remember we said like at the very beginning, if you had an amino acid chain of 10 amino acids long, right? How many different products could you make? And it was an insane number, right? Well, the same thing's true about once you have a particular sequence. Now, how can that sequence fold? Theoretically, there are lots of different ways, right? But there are only some folds that are productive, that make proteins that can do a function. So it's the same sort of limitation. And so you have to look at whether or not this protein sequence is going to fold. Well, that's all dependent upon energy, right? So Gibbs free energy, what is Gibbs free energy dependent upon? Equal to delta H minus T delta S, right? Okay, so entropy and enthalpy, these are two things that we kind of have to think about, right? So when you think about entropy, right? Why would it be more favorable to fold a protein? If you fold a protein, to me, what's happening to that disorder? You're getting less disorder. How is that favorable? Where do you see an increase in disorder when a protein folds? In the water all around it, right? Due to the hydrophobic effect, right? You have less of that protein exposed that doesn't like to hang out with the water. So the water does not have to arrange in this cage around all of these residues, right? You can fold up that protein and now we have a smaller space. And so you have to have less water organized around it. So you actually do, if you look at the whole thing, the system and the surroundings, then you're going to see an increase in entropy, okay? So most of the time, the equilibrium is in favor of the folded protein within cellular conditions. Is that the same thing as conditions inside of a test tube? No. So a lot of times you'll hear people have purified this protein and they want to look at its shape, and so they want to do X-ray crystallography. They want to do NMR. And they have to get a certain amount of this folded protein. Well, are the conditions inside of the test tube that you just purified this protein in the same thing as what was in the cell? No, not necessarily. So maybe in the test tube, it's not going to fold like it should. So you have to think about what free energy is going to be there in the cell versus the test tube. So a lot of times the in vivo folding process is more favorable because of concentration, right? You see the pictures inside the cell of all the proteins of everything. It's so compact inside the cell. The concentration inside the cell is about 30 times what we see in a test tube, 30 times in vitro conditions. So a lot of times we just can't get that concentration high enough in order to make that happen. The other thing that helps cells to properly fold proteins are other proteins that we call chaperone proteins. So chaperones form basically a surface for proteins to fold on. So these have a surface for proteins to fold on. Okay, it looks like we are out of time. So we will finish 4 next time. So what I'll do is I'll go in and I'll extend the homework 1 more class period. Okay. Questions? Good. Don't forget to turn in your exams. And I will add 2 points. Y'all have a good rest of your Wednesday. Yeah, anywhere. It's fine. Thank you. Anywhere. I'll take them so y'all can get out of here. Make it quick. Thank you, thank you, thank you, thank you. Awesome. Anybody else have any math errors or anything on their exam? Good.
I have a question.
Sure. Watch, you want to move out the way over here so that everybody can. I don't want to impede the process. We have to sign into plastic. Send me an email and I'll fix it for you. Yeah, sure. Thank you. Yeah. No, on the exam portion. The zip is the multiple choice portion.
Oh, okay. So there's the only kind of 10.
Yeah, no. Here. So this is all the stuff that I've hand graded on the exam. So it's these, the circle numbers at the bottom.
And to come see you, I just have to put in on your calendar.
Yeah, I mean, you can swing by the office if I'm there, but if you want to make sure, if you make the appointment, I will be there.
All right.
Yeah,
Thank you.
sure, sure.
Okay, so you're gonna take off the phone.
Well, yeah, I already did.
Okay,
Do you still see it?
Still showing us. 2 out of 3 or 1 out of 2.
I'm gonna go back and check because I pulled it from everybody, so it shouldn't be
Whenever I
showing
come
up.
back, it was still showing that I was.
Well, if I have to override it, I will.
Okay.
But if you're
I'll
having
check
that
it
problem,
again.
then everybody else is. Yeah, maybe just like shut it down completely
Okay.
and then restart it, and maybe then it'll communicate. And then send me an email, regardless of, hey, I checked, it's gone, or I checked, it's still there.
Right.
And then I can go from there.
Okay. And can I ask you
Sure.
something? Okay. So phosphate group and I transfer and I create ADP.
No. The transfer of phosphate from ATP to another molecule. So
how
we're
do I
putting it
know-
on another
How do
molecule.
I know what the intermediate is in that case?
It doesn't matter what the intermediate is, but it's highly reactive. That's the whole point.
But wouldn't you have a lower reactive if you lose a phosphate group for an ADP? Wouldn't you
No,
have a reactive?
but you are putting, it's not about what's left when you pull off the phosphate. It's about where that phosphate is going. Once you tag that extra phosphate group onto your new molecule that you're phosphorylating, basically, then this is very, very high energy.
So what is ADP? Is ADP not an intermediate in that case? Because it says, that's how I was
It's a
looking.
product.
But wouldn't it be an intermediate also?
No, because what the group, or the protein that the phosphate group has taken up doesn't hang out for a really long time. It's a very, very quick turnover. So it's an intermediate, and then it forms a more stable product. So then you get this more stable product, and you get ADP as a product.
So ADP is not here.
No, this is like ATP.
Like I phosphorylate some of ADP now and that.
ADP is a product. ADP is always going to be a product. It's not an intermediate. It's a product.
So, okay, you were 12. Okay.
Under what conditions?
This is
Yeah,
a-
it should have said, under
However,
what conditions?
in this condition, actually, so if G is less than 0, we have a spontaneous, which goes to the product.
So it is positive to go to the reactant.
I
So
got this
we'll go
right.
to the products. We'll
Okay.
go to the products.
So if I have a G so I'm going spontaneous to the reactant,
You're going spontaneous
towards the
to,
reactant.
yes,
So if I
spontaneous
was
to the reactant.
spontaneous towards the reactant, I would have to add more products to make it more spontaneous to the reactant.
Wait.
I think you were actually,
Products,
I don't
reactants.
want to like take
No,
people's points
it's
away.
fine. No,
but
I'm
but
gonna
I
give
was-
everybody the point. It's fine.
okay,
So
so
if I'm going
Spontaneous.
spontaneous,
towards
so
the
this is
reactant.
delta G positive to the reactant. No, to the, this way. So then
I'm
it
trying
is
to
delta
proceed.
G negative to the product.
So if I wanted to continue to go under what the reaction continue, like spontaneously proceed towards the reactant, you would probably continuous add products.
You would.
So it should be
So it
C,
should be C. Yeah,
but you had put,
but I had put B.
because I
I'm
actually
just
read it.
giving
the way-
everybody. Yeah.
Okay, because whenever you said that, I was like, wait a second, I think you might actually.
That's the point.
And also, last thing. Okay, if I say like, if I don't know something and I write it, is that worse than,
Yes. Yes.
because I thought I may have been right, but I was like, well, maybe.
No, but stick with what you know. Stick with what you
Because I don't
know.
want you to say, I don't want you to put, well, you should have put this as well, so it's still more. So I got, I have -2 for writing this as well.
As
not
well
writing-
as that, yep.
So had I not written this,
It would have been -1 because it contains uracil, right? There's no way of
you didn't put
telling.
-2, you said all of this.
I'm putting -2 for the whole problem. So uracil is deaminated, that's correct. Having tyrosine inserted instead allows DNA to determine if any mutation has occurred. Yes, so you said that, right, because we have cytosine instead of uracil, we can detect the mutations. So all you need to do is to explain how you get the mutation.
Okay.
Like why does this spontaneous deamination event lead to a mutation? So that's all you missed there.
So did you mark off points for
So
this or
I'm
not?
1 point for this and 1 point for that.
So had I not put this there, would it
One,
just been?
would-
Okay, so it's better. I just want to figure out how you.
well, if you tell me something that's wrong, like.
You know what I'm saying? If I wrote everything, even if I wrote this in here, and I didn't write that,
Then it
That's
would have been
all-
no points off.
that's all I want to know about how you grade.
Yeah, like if you tell me something that's wrong.
If I put 2 plus 2 is 5, you're like,
Yeah. Oh, thank you. Yeah, you too. What slide was that? I was supposed to mark where I leave off. We were almost done. Chaperones, yeah. I still have the Zoom going.